Hello, I'm Billy Rowell, a senior staff bioinformatics scientist at PacBio. Today I'll walk you through PacBio's consolidated HiFi human whole genome sequencing pipeline, a comprehensive streamlined analysis solution designed to harness the full power of HiFi data for variant discovery. PacBio offers two powerful platforms for HiFi sequencing. Our Vega benchtop system brings HiFi within reach of any lab, processing about 200 genomes per year. For production scale operations, the Revio system with Spark chemistry pushes the boundaries of what's possible, handling up to 2,500 genomes annually. Both systems deliver the same exceptional HiFi data quality at 20x coverage. Not all sequencing data provides the same insights. While short-read sequencing can detect single nucleotide polymorphisms in small indels, it misses critical genomic information. HiFi WGS delivers a comprehensive view of the genome, capturing structural variants, segmental duplications, tandem repeats, and enabling full genome phasing. Importantly, HiFi illuminates over 400 medically relevant genes in dark regions that are completely inaccessible to short reads. Plus, with HiFi, you get methylation detection with no additional prep or sequencing required. It comes standard with every run. At PacBio, we have developed or collaborated on a variety of tools to make the most of HiFi whole genome sequencing data. Each tool addresses specific variant types: PBMM2 for alignment, DeepVariant for small variants, Sawfish for structural variants, Target for tandem repeats, Paraphase for segmental duplications, and more. These tools are freely available through GitHub and Bioconda, with SmartLink providing a user-friendly interface. Effectively creating a comprehensive view of the genome requires integrating all of these tools. The WGS variant pipeline solves this integration challenge. It's a streamlined workflow that provides a single input point, implements best practices automatically, and generates integrated outputs. This lets you focus on biological discovery rather than computational plumbing. The pipeline is implemented in WDL, or Workflow Description Language, making it portable across compute environments. All dependencies are containerized for reproducibility. It runs both on-prem and in the cloud, with typical analysis completing in under 12 hours, depending on resource availability. While command line execution is standard, several partners offer point-and-click interfaces. The pipeline workflow is straightforward. You provide unaligned HiFi BAM files at 10x to 40x coverage, along with basic sample metadata, like the sample identifier and optionally the sex, the phenotype, and the relationships between samples in a family. The pipeline then generates comprehensive outputs, phased alignments with methylation probabilities, variant calls across all variant types, methylation tracks for each haplotype, pharmacogenomic diplotypes, and summary statistics. Everything you need for downstream analysis in one integrated package. The pipeline begins with alignments to GRCH38 with PBMM2, a Minimap2 wrapper designed specifically for HiFi data and file formats. Alignment parameter presets have been optimized to work with downstream variant calling tools. For calling single nucleotide variants and small insertions and deletions, we apply DeepVariant, a germline small variant caller developed by Google. We collaborate with Google to maintain and improve the DeepVariant PacBio model. And for analysis of multiple related samples, we joint call from DeepVariant VCFs with GLnexus, a scalable merging and joint calling tool developed by DNAnexus, with optimizations for DeepVariant inputs. Sawfish is a joint structural variant and copy number variant caller from mapped HiFi reads. It discovers germline structural variants from local sequence assembly and jointly genotypes these variants across multiple samples. Sawfish additionally applies copy number segmentation on each sample's sequencing coverage levels, synchronizing structural variant breakpoints with copy number change boundaries in the process to improve classification of breakpoint-based structural variant calls, in addition to calling copy number variants. Target is a tool for genotyping of tandem repeats from PacBio HiFi data. In addition to basic size genotyping, Target profiles sequence composition, mosaicism, and CpG methylation of each analyzed repeat and visualizes the reads overlapping the repeats. Many medically relevant genes fall into dark regions where variant calling is limited due to high sequence homology with paralogs or pseudogenes. Paraphase resolves these challenging regions, determining copy number and making phased variant calls. For mitochondrial analysis, MITRE-SAW removes nuclear insertions, realigns reads, and identifies both homoplasmic and heteroplasmic variants. HiPhase uses our highly accurate HiFi reads to jointly phase variant calls from DeepVariant, Sawfish, and Target into longer haplotype blocks that can aid in the interpretation of variants. This phase information is also applied to the reads to produce phased, haplotagged alignments that aid visualization in IGV. StarPhase is a phase-aware diplotyper for most CPIC pharmacogenomic genes that produces output that can further be annotated by PharmCAT to generate reports. And finally, the per-read 5M-CpG methylation calls generated on the Vega and Revio are processed by pb-CpG-tools to generate site methylation probabilities, including the probabilities for each haplotype when reads are haplotagged. The site probabilities are reported as both bed files and bigwig file outputs. Deployment is flexible for your infrastructure. On-prem users choose a Whittle engine like Cromwell or MiniWhittle and a container runtime like Docker or Singularity. They configure it for their scheduler, download the workflow and references, create a file describing their inputs, and launch. Cloud users select a platform like AWS HealthOmics, Azure, or GCP, copy the workflow and data to their environment, create a file describing the inputs, and launch. We also have partners offering managed solutions. The pipeline is already making an impact on research. Recent publications demonstrate its use in characterizing variation in large multi-generational pedigrees and in identifying variants in rare developmental and neurological diseases. These studies showcase the pipeline's ability to detect variants that would be missed by other technologies. Visit our GitHub for the complete workflow code. We provide demo data for testing, comprehensive documentation, and links to our compatible partners for additional deployment options above. PacBio HiFi whole genome sequencing and the WGS variant pipeline represent a significant advance in making comprehensive genome analysis accessible and reproducible. Feel free to reach out with any questions at the email shown, and I look forward to seeing how you'll use these tools to advance your research.